Real quick video over chromosomal mutations. Okay, so what you see on the screen right now are five different types of chromosomal mutations. And these tend to happen due to errors during crossing over. So as a reminder, during prophase 1 of metaphase 1 of meiosis, homologous chromosome pairs, they line up, they get together and form little tetrads where there's two replicated chromosomes, those homologous chromosomes that have the same genes in the same location, and they can swap bits, which is totally fine because they're swapping like for like. They're swapping, the same locus of the same chromosome for the same locus on the homologous chromosome pair. And so you're just trading one gene for another, or 6 of these genes for those 6 genes, and it's all fine. But sometimes you can have errors happen during that process, and that's how you can get some of these chromosomal mutations, where you could have an entire section of a chromosome go missing, what we call a deletion. This is more than just a deletion like for a single gene, like we talked about before, right? We already covered like insertion and deletions on a small scale within a single gene. For this one, we're talking about like an entire section of a chromosome, maybe with dozens or hundreds of genes being missing due to that deletion. Likewise, you could have something called a duplication, where instead of part of a chromosome going missing, part of a chromosome winds up getting duplicated. So instead of giving a part of a chromosome away, right, and then getting the same chunk back, maybe this chromosome just keeps both pieces, and this one gave it away, right? So this can often happen where a deletion and a duplication happen. One chromosome gets a section deleted because it gave its chunk to the other chromosome, but it didn't get one back in return, right? So you can have what's called a duplication, where you actually have all of these genes are duplicated, and there's actually 2 copies of all those genes now in this chromosome. You can also have something called an inversion, where a sequence of DNA is simply swapped around. These sometimes don't really have much of an effect at all. The genetic information is often still there, but depending on where the breakpoints are when this is inverted, you could have some genes get messed up. But generally, these aren't going to have as significant of an impact as some of the other ones, right? And so sometimes some chromosomes can get twisted up on themselves and wind up kind of crossing over with themselves and swap some parts around. You can also have what's called an insertion. Now, insertions and translocations down here, these are when you actually have crossing over happen between non-homologous chromosomes, which is not supposed to happen, right? So when those chromosomes pair up during that prophase 1 of meiosis 1, right, and do crossing over, they're supposed to be lined up with their homologous partner, right, the chromosome that has the same genetic information on it. But sometimes you can have them line up and swap parts with chromosomes that do not have the same genetic information on it, right? Like, for example, chromosome 5 pairs up with chromosome 8, and they swap parts, right? And that's where you can get things like insertions, where part of a chromosome that's not even that chromosome winds up being part of this chromosome here. That's an insertion here. Or translocation, where they actually like swap parts here, right? So translocation, this is actually exactly how crossing over is supposed to happen, but it's happening between non-homologous chromosomes, so that's bad. So now both these chromosomes are kind of like hybrid chromosomes, where this green one's got part of this red one in it. This red one's got a big chunk of the green one on it, right? And so these now no longer match any particular chromosome. And so all of these issues can result in other conditions, diseases, chromosomal abnormalities that lead to various challenges. But some of them, like I said, there are some insertions where it doesn't really have much of an impact. It all just really depends on the karyotype of the individual and what genes they have extra copies of, what genes they are missing copies of, because having extra or missing genes can obviously influence somebody's health. All right, once again, these are because of errors during crossing over during prophase 1 of meiosis.